begin this. Okay, so movement. Now, the main part of movement is the structure of muscles and sarcomeres. So the first is the skeletal muscles. The only part of muscles that we really need to know in the IB is skeletal muscles. We don't care about muscle cells elsewhere, basically. So skeletal muscles are attached to bones, and they're what cause movements of animal bodies. So they contain large multinucleated cells called muscle fibers. So as you can see here, there are multiple muscle fibers, which are called and then within each this specifically is a muscle fiber. Within each muscle fiber, there are small cylindrical structures called myofibrils, and around these are a special type of endoplasmic reticulum, which is also called the sarcoplasmic reticulum. So these bands are this that's surrounding it is a sarcoplasmic reticulum, and then the membrane at the outermost layer is also called the sarcolemma, or the membrane of the muscle fiber. Now, myofibrils contain of repeating units called sarcomeres, which have both dark and light bands. So as you can see, there's light bands, dark bands, light bands, dark bands, and it just repeats throughout the myofibrils. So the light and dark bands extend across all myofibrils in a muscle fiber, giving it a striated appearance, which is why it's white, black, white, black, white, black. And then in the context of this picture, and each sarcomere is able to contract and exert force, which is what allows for movement. So this is a sarcomere. I want everyone to try, it's okay if you don't get all of them, try to label A to E. And again, spoiler your answers if you're going to try to answer it. If you're not going to try to answer it, it's okay with me, I don't mind, but I would appreciate if people try to answer, if they remember. Is this correct? Sorry. Yes, I believe that was correct. Law, that was correct. It's not morning for me. It's nighttime, but it's been good. It's been good. Thanks for asking. It's been good. Because I've been prepping as well, because I have bio tomorrow. And then also geo. But no, it's okay. Don't worry about it. Yeah. It's like nighttime here now already, which is weird. But yeah, it's been good. It's like nearly dinner time for me at this point. I see all the question marks that people have put, and don't worry that you don't know, because it's okay, because I'm going to go through it. It's good that you don't know now, but then you'll know it later, which is why I wanted to check. So you'll have a higher chance of remembering if you make the mistake first and then relearn it, than if I just said it and then it wouldn't stick in your brain. It's okay, it happens sometimes. Okay. I think Steph is still answering, so I'll just wait. No, but I'm glad you did this now, so then there's a lesser chance of it happening in the exam. I think there's one more person who's still typing, so I'm just gonna wait until. Yes, exactly. It shouldn't happen on the exam now that you've done it. Okay. I think a lot of people got it right, even if they don't think they got it right, or if they don't know. So, okay. I'm gonna assume that most people are done. Yes? Good? Are we good? Yeah. I'm gonna assume that most people are done. So, we have. So, sorry, I didn't make it a bigger one, but so A was Z line because these are the Z lines. So at either end of a sarcomere, this one section is a sarcomere. At either end of a sarcomere, there is a Z line to which actin filaments are added. So D was actin filaments, the thin actin filaments. B is sarcoplasmic reticulum. It's not skeletal structures, it's sarcoplasmic reticulum. This sarcoplasmic reticulum is the thing that is a specialized type of endoplasmic reticulum. And then E was myosin heads and C was myosin filaments. Are we good? Yes? Z disc and Z line. I don't actually know. I've always said Z line, so I don't know if it would be considered the same thing. Not sure. Okay, so at either end of a sarcomere is a Z line to which actin filaments are fixed and attached. The actin filaments stretch inwards towards the center of the sarcomere. So they stretch this way in this direction, or for this one in this direction, towards the center, essentially, of one sarcomere. Between each layer of actin, there's a layer of thicker myosin filaments, which have the heads, which are the small projections here, that will form cross bridges by binding to the actin. Now, those cross bridges are what allow for the contraction to happen, and we'll discuss that in the next step. So, the part of the sarcomere that contains myosin, so this section is the dark band, and then the light band is the part containing only actin filaments. So everything past this and just before the end of the myosin is the dark band, and the smaller sections are the light band, which is why when you look here, the white bands are only in small sections, and they're in sections of 2 because that one line is the Z line, and then basically that's the Z line, and then the bigger is the myosin, and then actin, and the myosin, and then actin. So that's why it's bigger sections and then smaller sections of light band. Bigger sections of dark band and smaller sections of light band. Are we good with sarcomeres? I don't mind Krebs cycle and Calvin cycle and all of that now at this point. Okay, are we good with sarcomeres? Yes, yes. Yay. Okay, so sliding filaments and contraction. So the contraction of any skeletal muscle is achieved by the sliding of these actin filaments across these myosin filaments over each other. So the contraction will pull the ends of sarcomeres together, making the entire muscle shorter. So contraction is basically making the muscle shorter, but since this is one end of the sarcomere and this is the other end, these two ends will be pulled together to pull them closer and make the entire sarcomere shorter, which therefore in turn in the bigger scale will make the entire muscle shorter. The sliding of the filaments is an active process, and we'll discuss how it works, but they require energy from ATP. So the hydrolysis of one molecule of ATP will provide enough energy for a myosin filament to slide a very small distance across an actin filament. So it won't be very big movements with just one ATP. Therefore, a repeated cycle of events is required to contract muscle sufficiently enough to move in any way. So for it to get from here to this one is going to take a long time, because one ATP will only move it by a little bit, and then a little bit more, and then a little bit more. So there has to be a repeated cycle of events to take place. So it doesn't take a long time in humans anymore because of how fast it works. But if you think about it in terms of if we had only singular supplies of ATP, it would move very slowly. So here's the mechanism of muscle contraction. This I've taken basically from the entirety of the Oxford book, but there's basically 5 steps that go around. Now, in these 5 steps, you need to know about the ATP, about the myosin heads, and about the entire movement process of it all. So the first is myosin filaments have heads, which form cross bridges when they are attached to binding sites on actin filaments. So we know, based on this image, this is a myosin filament, these are the heads. So when we go to the mechanism, this is the myosin and this is the actin. Now this head, actually, am I right? Yes, I should be. This is the myosin and this is the actin. Yes, guys? Am I right? Are we good? Am I right? Yes. Yes. Actin is the twisty thing. Actin doesn't have a head, myosin has the head. Yes, which makes sense. Okay, so this base is the myosin and this is the actin. And so the myosin basically is fixed in one place, but it causes the actin to move, which is why this is the actin and this is the myosin. My bad. So myosin filaments have heads. This is the head that forms cross bridges with the actin, and then where they're attached to the binding sites. So this is a binding site, for example. The second step is when ATP will come and bind to the myosin head and cause them to break the bridge that had been formed. So as you can see here, it's linked together because of the cross bridge. Here it unlinks and breaks down the cross bridge because the ATP will come and bind to the myosin. And then ATP will be hydrolyzed to ADP and phosphate, which causes these heads to change the angle, and the heads will then be cocked in their new position as they're storing potential energy from ATP. Because when ATP breaks down, is hydrolyzed from ADP to when ATP is hydrolyzed into ADP and a phosphate molecule, energy is released, and that energy is stored in the head, and that's why it's in a cocked position. This the heads then attach to the next binding sites on the actin that are further from the center of the sarcomere than the previous sites. So for example, if the site was originally here, now it will bind somewhere over here. So when it moves and binds to the one further away, the ADP is released, and then the head will basically move from its cocked position back to its original position and push in this direction, which will then cause the movement of the actin filament to move towards this direction. So then when the ADP is removed, the heads will push the actin filament inwards towards the center of the sarcomere, and that's the power stroke. Now this repeated movement is what will lead to the to this happening, where both of the Z lines and the actin filaments will move closer and closer to the center of the sarcomere. Do we all understand the muscle contraction? Yes. Is troponin or on tropomyosin, or am I stupid? We'll talk about that literally next. The next thing we're talking about is troponin and tropomyosin. So I'll go through that when I'm talking about that. But are there any questions related to this muscle contraction movement? Because that's fairly difficult to pull off. Right, okay. So if you think about it, here, let's think about it in terms of this, yes? So what happens is, I'm gonna zoom in a little bit more. I don't know how close you can see that actually, because it's not that zoomed in. So annoying. Okay, hold on. Let me just insert a random thing here. Make this bigger. Oh, no, don't want to cut out the Z line. Okay. Let me just zoom this in as much as I can, and then I'll explain. So, if you think about it in terms of this structure, this is a Z line, this is a Z line. We know that this is the myosin with its heads, and this is the actin. So if we look at just this, let's look at here. This myosin head has formed cross bridges with the actin filament here. But what happens is that the cross bridge between this specific portion and this head breaks when ATP is joined. And then when ATP hydrolyzes and becomes ADP, this head will gain the energy and therefore move, caulk its way here. So it'll become not in this conformation. It'll be stood up more straight. Even though everything is in the same position, it'll be stood up more straight. And then we'll form cross bridges with this portion. Instead of the portion here, it will now form cross bridges here. So then that will... Actually, does that make sense? No, that's the reverse direction. Sorry. Instead of making... Is that right? Sorry. It's harder when it's in this kind of conformation. But if it goes here, which pushes it that way. No, so once it's caulked, it will basically form cross bridges instead of here, further here. And then when the ADP that's joined here will leave, when it leaves, since it's joined here, this will go from its further slanted position back to this conformation. And since it's linked here, when it goes back to this conformation, since this myosin does not move at all, this will be linked to that. And then once it returns back, this portion will move in this direction to be able to adjust to where the myosin is. This is a lot more faster than it should be, but does it make sense? So it attaches to the actin and moves it. Yes. That's what happens here. So it's usually already attached, but then it detaches, attaches to another portion, and then moves it. Okay. Are we good? When actin and myosin head attach, does the actin move left or right? That depends. Are we talking about the actin here or the actin here? If it's here, this actin moves, basically it moves towards the center of the sarcomere. So if it's this actin, it moves towards the right. If it's this actin, it moves towards the left. And all the processes happen at the same time, which means that these will all be synced up to act at the same time, and then this will move as one singular unit. towards the center. So if this is the theoretical center, this will move towards this will move towards this. Which is why it's contraction, because it moves closer together. So it'll go from this to this, because this has moved this way and this has moved this way. So if you look at the left side of the myosin filament, okay, so myosin doesn't move, first of all. But if we look at the left side of the myosin filament, then the actin will move towards the right. Yes. If we look at just this section, these four thin actin filaments will move towards the right. The head moves, but myosin stays in place. The myosin doesn't move. The myosin head moves. The image with ATP and ADP shows the actin filament moving left. No, it doesn't. Does it? Why does it? Okay, yeah, because sorry. It's because I've been explaining on two different things. This is for this side of the situation. So this would be when this is a right actin filament, so it will move further this way. That's my bad, because it's just two different orientations. Okay, I'm gonna assume we're good and then keep going. So we went through this. Control of muscle contraction. So when a motor neuron stimulates a striated muscle fiber, so that's how muscle contraction happens, by the way. A motor neuron will stimulate a muscle fiber and therefore release calcium ions from the sarcoplasmic reticulum. So we know that, let me go back. This is the sarcoplasmic reticulum. This is what will let go of calcium ions or release calcium ions from the sarcoplasmic reticulum. This calcium will then bind to troponin, which is a protein that is associated with the actin filaments. So with these guys. The calcium will cause the shape of the troponin to change and then cause the movement of tropomyosin, which is another protein that is associated with actin, which will expose the binding sites on actin. So that's why myosin heads can form cross bridges by binding to actin. So actin has troponin and tropomyosin associated with it, right? There's two proteins that are associated with this actin filament. There's troponin and tropomyosin. When calcium is released by sarcoplasmic reticulum, it changes the conformation of the troponin and moves tropomyosin, exposing the binding sites. So when we look here, these binding sites are only exposed after tropomyosin has been moved because troponin's conformation has been changed. To investigate the control of muscle contraction, we have tried to use radioactive calcium or calcium-45, and so using autoradiography, we could find that radioactive calcium is concentrated in the region of overlap between actin and myosin filaments, but not in relaxed muscle. So when in this region, up until here, this region is concentrated in calcium-45, but this region is not concentrated in calcium-45 because the calcium-45 is required for the control of this process, but is not required in the relaxed area. This is because calcium ions are bound to troponin, allowing the cross bridge formation and sliding of filaments. Are we good? I'm gonna assume silence means good. Okay, so Now, movement. So, body movement requires muscles for us, and as muscles will only exert force when they contract and not when they relax. A muscle can only cause movement in one direction, but for opposite movements there has to be a pair of muscles that exert force in opposite directions, which means there has to be an antagonistic pair of muscles. So that's why always when we have, for example, an elbow, there is one section of muscles that will contract and that will relax for one movement. Muscles are typically elongated structures with tendons that form attachments at both ends. So tendons link muscle to muscle. One end of the muscle is the anchorage, which is a firm point which is a firm point of attachment that does not move when the muscle contracts. So in humans and other vertebrates, bones are the anchorages, but in insects and other arthropods, the exoskeleton will be the anchorage. So for us, one end of the muscle is the anchorage and the opposite end is the insertion. But in us, in our case, it's bones are both of them. So muscle contractions cause the bone to move together with its surrounding tissues, and therefore bones and exoskeletons act as levers, as they can change the size and direction of the force exerted by a muscle. I will ping you when we go to placenta, yes. You're trying to do another video and catch up with us? I mean, we're going to go to the elbow next. I'm just doing it in an unconventional order. Okay. Are we good with movement? Does it make sense? The antagonistic pair? Okay. Someone's typing, so I'm going to wait. What units have I gone over already? I've gone through 6.6. I've gone through 11.1, and now we're at 11.2, and then I'll go through 11.4 and 9 and C, or as much of everything as I can. Yeah, no worries. I don't do option A, sorry. I only do option C, which is my bad. But yeah. Okay. So in junctions between bones, so between a bone and another bone, when they meet, those junctions are called joints. Some joints are fixed, such as the joints in our head. So if I go all the way back, these joints between in our skull, these joints are fixed joints. There's no movement here, and those are the fixed joints. But most, oh my god, move! Why? Oh yeah, sorry. So most of the joints between the plates of bone in our skull are fixed, but other joints allow movement or articulation. So most of the joints that allow movement are synovial joints, which means that they have three main parts: cartilage, synovial fluid, and joint capsule. So cartilage is the is soft bone, essentially, that covers the surface of the bones to reduce the friction where they could rub against each other. Synovial fluid is between the cartilage covered surfaces to lubricate the joint and further reduce friction. And then joint capsule is the thing that seals the joint and holds in the synovial fluid within our joints. So we need to know what ligaments are and what tendons are. So ligaments connect the bones on opposite sides of a joint. So ligaments are bone to bone, and tendons are bone to muscle or muscle to muscle usually. So tough cords of tissue that connect the bones on opposite sides of a joint are known as ligaments. They restrict our movement and help to prevent dislocation. So for example, in our elbow or in our knee joints, there are hinge joints. So to prevent them from dislocating, ligaments are tough cords of tissue that will restrict how much you can move, so you can't end up dislocating your joint, basically. So ligaments ensure that certain movements can occur at a synovial joint but not others. So for example, our elbow has a lot of movement in one plane, so you can bend it, but you can't move it, like you can't move it in a 360 direction, essentially. Bolo? हाँ, दे देना, दे देना. Yeah. Does that make sense? This is 11.2. Which one allows synovial allows movements, and fixed or don't allow movement. The joints, yes. Synovial joints allow movement of just movement in general, and fixed joints, there's no movement at all. Fixed joints are most just for us, it's mostly just in the head, in our skull, but we don't need to know. Okay, so the last bit is I want everyone to label from A to I. This is the elbow. This is the last bit of this of 11.2, and then we'll go into the hardest part, reproductive system. Oh, and please spoiler your answers. So add the double bar on either side if you're gonna answer. That's the answer. Okay, I'm gonna stop screen share for a second so I can fix the issue with this. Okay. Try to answer as many of them as you can. It's okay if you can't do all of them, because I will go through it anyway, but it's just good practice. No, I think you got everything right in terms of spelling, except cartilage, but that's okay. Yeah, there you go. Yeah, I think a lot of you are getting this right, which is good. Okay, I'll wait for Steph to finish because I think they're the only one still trying, and then I'll go into. Okay, so I think most of you have them right, but I'll go through all of them anyway because I'll also go through the functions. So A is the biceps, which is the flexor muscle, which is the thing that's used to bend the arm at the elbow. The B is the tendon, which attaches the muscle to the bone. C is the radius, which is the bone that is the insertion for the biceps and acts as a lever, which transmits forces from the biceps through the forearm. D is ulna, which is the bone that is the insertion for the triceps, not the biceps, for the triceps, and acts as a lever and transmitting forces from the biceps through the forearm. It's basically the same thing as the radius, but for the triceps instead of the biceps. E is cartilage, which is the layer of smooth and rough, tough tissue that covers the ends of the bones where they meet to reduce friction. So this is a synovial joint, of course. So that's the cartilage, and that's the cartilage. It basically covers the entire joint and where they meet. F is a synovial fluid. It lubricates the joint and reduces the friction and allows for ease of movement. G is the joint capsule. This basically encompasses the entire joint and keeps the cartilage and the synovial fluid in shape. H is the triceps, which is the extensor muscles. So there's the flexor and the extensor, which is used to straighten the arm. So the flexor is what gets contracted when we bend our elbow, and the triceps are what get contracted when you straighten the elbow. I is the humerus bone. This provides a firm anchorage for both biceps and triceps. I think almost everyone got all of them right, so we're good. This is the end of 11.2, I think. So we're good. Any questions? I'm going to stop this here.